Hi, everyone. Welcome to today's PacBio webinar, Filling the Gaps in Rare Disease Research with Long-Read Sequencing, Stories from Undiagnosed Cohorts. I'm your host and moderator today, Nina Gonzaludo. I'm at PacBio. Today, we are joined by our guest, Dr. Ramki Rajagopalan, Director of Translational Bioinformatics in the Division of Genomics Diagnostics at CHOP. Before we get started with our talks, I just wanted to go over a few webinar housekeeping items. So the presentations for today have been pre-recorded. Ramki and I will jump on at the end for live Q&A. We have 2 webinar sessions today. If you have any questions for either of us, please go ahead and submit them in the questions module, which is provided on your attendee control panel. We also have a number of pieces of PacBio literature available in the materials module. And on-demand recordings of this webinar will be shared and made available on pacb.com, as well as on our YouTube channel after the talk. And as with all of our webinars, please fill out our post-webinar survey. It really helps us to find material that's relevant to your research. So with that, we will kick it off with the recordings. Thanks. I want to start with something that I'm sure is very familiar to many of you working in rare disease, and that is the diagnostic odyssey. Despite advances in sequencing technologies, rare disease testing is still includes a long and complex journey. Families often move through a sequence of tests shown here. So that might be single gene assays, microarrays, panels, exomes, sometimes whole genomes. And each step is designed to answer a specific question, but no single test provides a complete picture. And each test is adding time and costs, but still only provides a partial answer most of the time. So each negative result leads to delays and uncertainty, which has a lot of impact on the families. And when you look at the full journey, this can take several years. And for labs, this can cost thousands of dollars in testing and analysis alone. And even after all of that effort, many cases remain unsolved with legacy technology. So the challenge is not just about generating more data, it's about generating the right data earlier in the process, something more complete than the fragmented tests in this current odyssey. HiFi sequencing is a great option for more complete testing. PacBio HiFi sequencing delivers long reads, typically up to 25 kilobases, which is around 100 times longer than short read NGS. And HiFi also has very high accuracy, as these reads come directly from native DNA with no sequencing bias, allowing for complete coverage of the genome. And with HiFi, you also get 5-MC methylation calling for free without any additional library prep. And we'll soon support 5-HMC as well. And what this unlocks is a reference or clinical quality genome. It allows for de novo or T2T assembly and comprehensive coverage of all variant classes. And these include variant types that are inaccessible or poorly mapped by short reads, including structural variants, seg-dupes, tandem repeat expansions, and long reads also enable phasing, which is really important for rare disease variant interpretation. So what does this enable in practice? Across recent studies, HiFi whole genomes have demonstrated strong performance in detecting variant types that are challenging to resolve with standard approaches, including those in clinically relevant regions. And several of these studies are actually described in our updated Rare Disease Solves white paper, which you can find in your literature module. But I will walk through a few of them here. So on the left is a study from our HiFi Solves EMEA network, which looks at data and analyses that were reproduced across 4 sites in Europe. And in this study, they focused on known complex pathogenic variants in 11 paralogous loci that typically require multiple assays to detect in standard of care. And they found that 73% of these variants could be automatically called with a standard HiFi analysis workflow, and the remaining could be called with paraphase, which is a specialized tool that we developed at PacBio for calling segmental duplication regions. So overall, they were able to detect 100% of the challenging variants in the study, and they conclude that HiFi is robust, reproducible, and capable of addressing the most challenging cases. The study on the right shows what this could look like in practice. This is a study out of Radboud University Medical Center in Nijmegen in the Netherlands. And here they looked at a large cohort of 1,000 samples designed to mirror standard of care workflows. And here they found that HiFi showed over 96.4% concordance, while also identifying additional findings that help refine genetic insights. And again, this study was not designed to maximize long-read discovery. Instead, they were benchmarking directly against standard of care using the same clinical hypotheses, meaning in many cases, HiFi analysis was restricted to candidate genes that legacy assays test for. But beyond the 96.4% of concordance, they found that HiFi improved or refined outcomes in 3.4% of cases. And this was largely through haplotype phasing and detection of variants that existing workflows couldn't resolve. So this study also highlighted that many current workflows still require multiple technologies and iterative testing, and HiFi offers a potential to consolidate those with one assay. This slide shows a similar study from Children's Mercy, Kansas City. And it's another strong example of how HiFi whole genome sequencing can impact rare disease workflows in practice. And here the authors compared HiFi whole genomes directly against standard of care again, testing in pediatric rare disease cases. And what they found was a significantly higher yield with HiFi sequencing, 37% compared to 26.9% with standard of care workflows. And similar to the last study, here the additional findings were driven by variant classes that benefited specifically from the capabilities of HiFi sequencing. So this includes aberrant methylation, rare repeat expansion disorders, phasing of single nucleotide variants in singleton cases, and detection or refinement of SVs. In the study, they also demonstrated a much faster and simpler workflow. So here they looked at average turnaround time from test order to report, which is shown on the left figure here. And they found that it was reduced from 62 days with standard of care testing down to 27 days with HiFi sequencing, largely because they don't need as much iterative testing. So again, this is a powerful example not only of increased genomic resolution, but how consolidating multiple testing modalities in HiFi can improve efficiency and decrease turnaround time. This study from GeneDx, again, highlights the shift towards assay consolidation that I've been talking about in rare disease genomics, moving from multiple sequential assays towards a single comprehensive workflow with potential for first-line testing. So in this study, they evaluated 191 real-world clinical rare disease samples that were enriched for difficult-to-detect pathogenic variants. And using HiFi whole genomes, they detected 99.6% of the 481 known pathogenic variants in the study, including, again, many of those that are traditionally challenging for standard workflows. They included SVs, repeat expansion, methylation abnormalities, and variants in highly homologous regions. And they also looked at the operational perspective here. So many of these samples had already required multiple orthogonal assays, shown on the figures on the left, again, including microarrays, panels, MLPA, Sanger, targeted approaches. And here, HiFi showed the potential to consolidate these into a single workflow while still maintaining very high clinical sensitivity. So while I spoke a lot about whole genome sequencing, PacBio systems enable a number of other applications on the same platform, making HiFi a great tool for multiomic applications. So beyond characterizing the full spectrum of genomic variants, using the Iso-Seq method with K'Nex kits, you can also get a complete view of the transcriptome as well. This can unlock discovery of RNA isoforms, fusions, and express mutations with full-length coverage of isoforms or transcripts. As I mentioned in the beginning, with HiFi, you get 5-MC methylation for free. And we also have 5-HMC calling coming soon with SPRQ-Nx chemistry, as well as 6-MA chromatin epigenetic information available with our FiberSeq protocol. And if you're only interested in certain genes, PacBio systems support targeted sequencing as well, either with Amplicon's hybrid capture or pure target, which is a Cas9-based amplification-free method that is really great for looking at repeat expansions while preserving methylation. So here's an example of how multiomic capabilities like methylation add value to HiFi sequencing in rare disease research. This study focuses on FSHD, which is a rare muscular dystrophy associated with the D4Z4 variable number tandem repeat. This is one of the most challenging disease-relevant regions of the genome. D4Z4 is large, repetitive, GC-rich, and highly homologous, which makes it really difficult to resolve with standard approaches. And using HiFi whole genome sequencing in a computational tool called Kiwi, which was developed by our bioinformatics scientist, Xiao Chen, we were able to report repeat size, chromosome of origin, methylation level for each D4Z4 allele, and disease-associated versus normal haplotypes. And in this study, Kiwi detected all contracted pathogenic range alleles and showed distinct methylation patterns, which you see on the right here, between FSHD1 and candidate FSHD2 samples. The key takeaway is that HiFi sequencing can bring together this repeat sizing, haplotyping, and methylation profiling all in one assay. And so for rare disease research, that gives you a complete view of both the genetic variant as well as its functional context. Another area where long reads are having a major impact in rare disease research is transcriptomics. Many rare disease cases are driven by splice disrupting variants, but these events can be extremely difficult to interpret with short-read RNA-seq. So in this study from the University of Southampton, researchers used PacBio K'Nex full-length RNA sequencing to better understand clinically relevant splicing events in rare disease samples. And with K'Nex, they were able to confirm 21 known splicing events and uncover additional transcript-level effects in 8 cases, including intron retention, exon skipping, isoform switching, and allele-specific expression. And in the study, more than 10% of the detected transcripts were novel and not present in standard transcript catalogs. And this really highlights how much transcript diversity can still be missed with conventional approaches. So the advantage of full-length RNA sequencing is that you are observing the complete transcript structures directly because they fit on a long read, rather than computationally reconstructing them from fragments like with short-read sequencing. And so for rare disease research, that more complete view of transcript biology can help explain disease mechanisms that would otherwise remain unsolved, all on the same platform that you would use for HiFi whole genome sequencing. One important point I want to emphasize is that generating comprehensive HiFi data is only part of the story. And to fully realize the value of long reads, you also need the right computational tools to interpret all of the variant classes present in the genome. So we have a PacBio whole genome sequencing variant pipeline. It integrates best-in-class tools like Paraphase and the Kiwi tool I mentioned into a streamlined analysis framework for HiFi whole genome sequencing. So instead of needing separate workflows for each variant class, this pipeline brings together all of these analysis tools into a single integrated framework. It's available as a WDL and also available on compatible partner platforms. Again, the goal here is not just to generate more data, but to make the comprehensive genome interpretation practical and accessible. And because these tools are integrated into SmartLink and cloud workflows and GitHub, as well as compatible partner ecosystems, you can adopt these best practice HiFi analysis tools without having to build all the components and stitch them together yourself. Finally, I want to wrap up talking about scale. So one of the biggest shifts happening in clinical genomics right now is cost and scalability cost dropping and scalability increasing. And with our newest SPRQ-Nx chemistry, which will be available on Revio later this month, HiFi genomes are expected to reach approximately $300 to $350 per 20x genome, making this the most affordable HiFi genome yet. That's about 30 to 40% less than today, and roughly 10 times cheaper than HiFi genomes were in 2022 when Revio first launched. What's important is that you can get these savings without changing your workflow. So with the same setup as before, SPRQ-Nx chemistry also enables richer multiomic data with 5-HMC calling, along with 5-MC and 6-MA methylation that's available today. And what's exciting with SPRQ-Nx is that we're also enabling reuse of SMRT cells, which happens on instrument, so no additional touch points needed. If you're interested in hearing more, we had a separate webinar on SPRQ-Nx data, and that's available on pacb.com. So this is really about enabling scale, and it makes comprehensive HiFi genomes and multiomic studies much more accessible for clinical and research workflows. So with that, I'm going to go ahead and hand it over to our guest speaker, Dr. Rajagopalan. We'll both jump on at the end for a live Q&A session, so please drop any questions you have in the Q&A module. Thank you.
Hello, everyone. My name is Ramki. I am an assistant professor in the Department School of Medicine, University of Pennsylvania. I'm also the director of translational bioinformatics in the Genomic Diagnostics Lab at CHOP and the CHOP Undiagnosed Diseases program. Today, I'm going to talk to you about filling the gaps in rare disease research with long reads, present you some cases from our experience using PacBio long read sequencing in resolving variants of uncertain significance or resolving diagnosis for previously undiagnosed patients. Just to give you a background, our hospital is an academic medical center. We have 2 hospital locations in Pennsylvania with 627 beds and 50 plus network locations in Pennsylvania and New Jersey. We have about 1.5 million outpatient visits. So we're a big hospital academic medical center. I am part of the Genomic Diagnostics Lab within CHOP. We're a full service clinical genomic lab offering tests for both oncology and inherited genetic disorders. We do everything from Sanger to classic cytogenetics to genome-wide molecular testing. I wear 2 hats here, one on the clinical side, thinking about clinical assays that we offer to our patients, and also the research side when we think about how to bridge the gap between research and the clinical application of these assays. So when we look at the big picture, there are 3 main goals for me. One, how do I detect all major class of variants in a single diagnostic assay? And I think this is very important from the operational perspective, and also, how the tiered testing system, current day tiered testing system works. On the other end is that I want to find all disease-causing variants, right? I want to be able to find every patient who is suspected to have a genetic disease. Can I find a diagnosis in them? Finally, the middle piece is the gap between the research and clinical labs. Usually, clinical labs are behind when you compare to research, and there are several reasons for that. And how do we bridge these two separate sides of the same thing? Before I begin talking about the research cases that I'm going to present today, I just wanted to briefly talk about like what makes something a clinical diagnostic test. We operate on a framework developed by the CDC. This is the foundational framework for making something diagnostic tests. It's called ACCE framework, talks about analytical validity, clinical utility, and ethical and legal social implications. Before we make anything a diagnostic test, we have to think about all these aspects of the diagnostic assay, and that's when it becomes a clinical test. When we talk about diagnostic assays, they come in different types of variants require different assays, and sometimes operationally, some of the regions in the genome that are inaccessible by one technology forces you to use multiple technologies today. We've come along this far, starting with multiple different testing for like Sanger sequencing to panels to exomes to copy number variants from the chromosome microarrays. And then today, most of the labs offer short-read genome sequencing with structural variant detection. We don't detect like all possible structural variants, but this is becoming the first-line standard care today. Still, it's not complete. We still use some of our tests use multiple assays to achieve completeness. For example, the standard care diagnostic assay for mitochondrial disorders today in our lab uses two different assays. First one is long-range PCR, followed by next-generation sequencing. This is to detect SNVs and indels, and this assay cannot detect the levels of heteroplasmy because the PCR is involved and the heteroplasmy levels are not reliable. For that matter, like we use droplet digital PCR. This quantifies the deletion heteroplasmy, but it cannot detect sequence variants. Similarly, more recently, I would say, 4 years ago, the test that we offer for non-syndromic hearing loss used multiple assays, like 6 different assays, for example, right? Non-syndromic hearing loss is a heterogeneous disorder with more than 120 genes known to be associated with it, and many genes require special assays for completeness. For example, STRC is a known gene with a pseudogene, and that requires long-range PCR. Then we don't get the copy number from the exome, so we do array CGH. So this kind of like increases the burden of testing in the lab, time taken to give the results back, time taken to achieve a definitive diagnosis. So this goes back to my slide talking about how it is interesting, how it is important to move to a single comprehensive diagnostic assay. And that's when long-read sequencing comes in. Long-read sequencing has the potential to become a single comprehensive assay. I'm not going to talk about the technical details of the PacBio sequencing to this crowd, but it checks all the boxes. For example, it gives like magnitudes longer reads. The error rates is as comparable to short reads that we have been used to. There's no amplification required for library prep, so that's a huge advantage. Like, we talked about the mitochondrial genome example. That the long reads offer better detection of large structural variants and variants in repetitive regions. And out of the box, you get methylation. And I think when we talk about multiomics, you get multiomics right out of the box. And then, of course, like the long reads enable you to face variants in cis or trans, right? And all the results that I'm going to talk today in this presentation is produced using PacBio HiFi long-read sequencing data. Before we get into the actual cases, I just wanted to talk about the difference between research and clinical applications because I do this on both sides. In research, most of the time we're looking at cohort level findings or shared events between individuals in the cohort. But in the clinical lab, like we're looking at one patient at a time. And this brings a lot of considerations when it comes to analysis. And like, how do you offer the same analysis protocol for every patient that walks into your lab? In research, most of the times, like we focus on like what is found right? But then in the clinical lab, we have an additional responsibility, added responsibility to say like what we ruled out. It's not just like what did we find, but it was also about like what was confidently ruled out. Then in the research, most of the times we're not reporting the results back to the patient. In the clinical labs, we do report results back to the patient, positive or negative or inconclusive. So there's a lot of liability that comes with it. So clinical setting requires highest quality standards when it comes to offering these tests. Now I'm going to talk to you about diagnostic odysseys. A lot of people today working in rare disease like talk about this. Diagnostic odysseys are long, challenging, and very expensive. On average, a patient goes through this for like 5 to 7 years, and they are families, of course. It is very expensive, about like $19,000, and 13 genetic tests. And in the way they get misdiagnosed 2-3 times, this is not uncommon as well. This is a huge burden for the family, for the patient, for the providers, and the payers. So now I'm going to talk to you about 2 different families with the same syndrome with the similar deletions they found, and just to demonstrate how different each patient's diagnostic odyssey is. Here's Dylan, who came to us in 2009 with congenital diaphragmatic hernia, and he got a SNP array, and he went on to develop other phenotypes like over the next 2 years, and he got multiple different panels tested, like with a clinical diagnosis of Smith-Magenis syndrome and Opitz-GBB syndrome, and then going on to get a clinical exome, which was negative, and then the research exome, which was negative. Logan, his brother, came in 2010, and he had a very different path to diagnosis. And he was, in fact, like he had a very different diagnosis. From the clinicians, he got an excellent panel testing done in 2014, which was also negative. But in 2015, they both got the same MET39 neurodevelopmental syndrome diagnosis informed by their exomes. Now onto another patient. This is a different patient with MET39 syndrome, the same syndrome that I talked to in the previous family. They came to us in 2003, and you can immediately appreciate the different kinds of testing and different clinical diagnoses they got over the years to arrive at the same diagnosis that the previous patient got. So each patient's diagnostic odyssey is different, and they go through multiple levels of testing, and this process is very tiresome and expensive. Just to stress on that, like here's data from the cohort of 894 subjects with birth defects. And you can appreciate this. The graphs on the right side shows the complexity and the tiered nature of testing. For example, like, more than 40% of the patients in this cohort had like more than 2 testing done. If you see this graph E, you can see like the combination of testing done, and that's very different across the cohort. It's not very consistent. So this is a huge burden. And I know the times are different today, but this is not uncommon. Even after exome became the first tier diagnostic test, if we see a patient today in our clinic, it's not uncommon for them to have like multiple different combinations of testing done over the years. Now that brings us to the research side of things. Like, where are these missing diagnoses, right? More than half of the patients do not get a diagnosis, right? The middle panel shows the diagnostic yields from literature. About like 40 to 45% of the patients get diagnosis from whole genome sequencing tests, and people can agree or disagree, or maybe there are different numbers, but like it's about that 40 to 45%. If you look at our lab, the diagnostic yield from our lab, it's consistent at like 20% for our exomes and genomes. This is quite different when you compare to what you see, because our patients, like when they come into our clinic, most of the times they have had prior testing done. And in contrast, like you can see our rapid sequencing for the NICU patients from 2024 shows like 31% diagnostic yield, most likely because that is their first diagnostic test. So with that, like I'm going to talk about a few cases from our undiagnosed cohorts. I'm going to talk about a cohort of patients with Cornelia de Lange syndrome. These patients went through standard of care sequencing and del-dup assays for those genes and exomes and genomes. And another cohort of patients from the CHOP undiagnosed diseases program. Here's a very neat example of detection of the small insertion and phasing in a patient from the undiagnosed diseases network. So this patient came to us at 18.5 months of age. They had global development delay, developmental regression with loss of previously acquired motor and social milestones. They had extensive diagnostic testing prior to enrollment in the UDN. But all of those testing were negative. When we did long-read sequencing, we were able to find a 98 base pair insertion in an exon in the gene LIG3 and a splice site variant in trans. The beauty of this is that the insertion is an interesting variant to detect because the insertion, the sensitivity to detect insertions using short reads goes down with the length of the insertion. And also, depending on the sequence complexity, you might have difficulty recognizing a true insertion versus noise. But long-read sequencing with PacBio, it's very nice and clean, and you can immediately recognize. And these variants were 8.5 kb apart, and you can immediately recognize that they're in trans just by phasing the long-read genome. Here's a patient example from the cohort, Cornelia de Lange syndrome cohort. This is a patient who went through standard of care sequencing for Cornelia de Lange syndrome diagnosis, which is sequencing and del-dup analysis of all the genes associated with. It's a dominant disorder. There are 6 genes known to it. 65% of the patients have mutations in the gene NIPBL. They were all negative. They went on to get a research exome and research genome, which were also negative. And when we did PacBio long-read sequencing, it was quite obvious to us that there's an insertion in the intron 22 of NIPBL. And we also were able to immediately recognize that it is an SVA element that is inserted into this intron. And I'm showing you the alignment here and the de novo assembly here. You can appreciate how clean this data looks and how you can really appreciate the mutation here. In the short trees, like we can see the insertion here, but it's non-specific and it's a repeat element. We cannot actually size the repeat element. Here you can immediately recognize the size of the insertion from the long read data. Here's another example from the Cornelia de Lange cohort. This patient also went through standard care sequencing and del-dup testing for the Cornelia de Lange and then research genome, and they were negative until we did long read sequencing and immediately recognized that this is also another repeated element insertion in the intron 32 of NIPBL. And you can clearly see how good this insertion is characterized, like when you see the de novo assembly, even in the alignments. And this is a harder one for us. Like when you look at the short trees, like in the previous example, you could see that the boundaries of the insertion, you still cannot size it. In this example, only one side of the insertion, it's a poly sequence, and you cannot recognize the other end of the insertion. But doing long-read sequencing gives you an immediate visibility into like what exactly it is and be able to characterize this insertion. Here's a third example of insertion from the Cornelia de Lange cohort. This is a dispersed duplication. This is quite different from the previous 2 insertions I showed. This is a 144 kb duplication from the chromosome 5, 34 megabase region of the chromosome 5 inserted into NIPBL. This is a huge insertion. And This is quite different because of the length of the insertion, and also you need. I don't have short reads for this. Like you need you need to put multiple information together to understand this insertion. If you were to only depend on the short reads, in the long reads, if you can see, you can readily appreciate the sequences from the stop clip, and also you can when you do the de novo assembly, it's much cleaner. And you can appreciate the insertion immediately. Now that brings me to talk about the multiomics. Multiomics is the buzzword today, and we're getting started. And when we think about multiomics, I think the long read sequencing enables you to do multiomics right out of the box, because you get methylation data right out of the box, and that's multiomics, right? So I'm going to show you a couple of examples from our undiagnosed diseases cohort, how we used PacBio long read sequencing to identify variants that were previously unrecognized, and also characterize and resolve uncertain variants from prior testing. Here's a patient in the CHOP Undiagnosed Diseases program. She came to us in 2019. She had a prenatal diagnosis of skeletal dysplasia and Joanne thoracic dysplasia. She had SNP array fish and karyotype prior to enrolling in the UDP. In 2019, she had an exome and skeletal dysplasia delta panel, and then another panel in 2020. Mostly due to the coverage of the previous panel, gene coverage in the previous panel, and then a whole genome in 2021. All of them were negative, right? She clearly has a very specific phenotype, but we were not able to pinpoint a variant molecular diagnosis for her. Then in 2022, we sent EpiSign, which came back positive with a diagnosis of Kagami-Ogata syndrome, right? So Kagami-Ogata syndrome is an imprinting disorder originating from chromosome 14, and majority of the cases are UPD14 cases. There are two syndromes associated with this imprinted region: Temple syndrome, which is paternally methylated and maternally expressed, and Kagami-Ogata syndrome, which is maternally expressed on the maternally expressed allele and paternally silenced. That point in time, we were thinking, do we do additional diagnosis to clarify this? And SNP array was negative. It was not UPD. Both the haplotypes were from different parents. So we were thinking, what additional diagnosis or what additional testing should we do? We performed PacBio long read sequencing, and we found a 203 base pair de novo deletion in the exon 1 of MEC3. This is the maternally expressed gene in the imprinted region of chromosome 14, and we were able to find this deletion and phase it with a paternally inherited SNB that was 1 kilobase away. This is very interesting finding, because the 203 base pair deletion falls in the gray area of, I would say, the variant detection sensitivity in the short reads. And also, depending on the sequencing context, again, as I said before, you could have, it could be challenging to identify these insertion. But long read sequencing, you can have confident variant call of this size. So now I'm showing you an image that shows the methylation data from the methylation microarray in the middle panel, and then the long read sequencing data on the bottom panel. Here's the variant that we found, and we were able to phase the variant that this deletion is actually occurring on the maternal allele, right? Here's the exon 1. And this middle panel shows you the methylation microarray. The orange lines show the patient data, solid lines, and the dotted lines show the control data. The orange color shows the maternal allele, and the blue one shows the paternal allele. The y-axis is the modification probability, or you can call it like methylation, methylated, percentage methylated, that locus. And you can see that in the control data set, the paternal allele is methylated and maternal allele is unmethylated or hypomethylated. In our patient data set, you could see that both maternal and paternal allele is hypermethylated. So this is additional testing, right? Like this is from the methylation microarray. Whereas in the long-read sequencing data, this is the methylation detection from the long-read sequencing data. And the top one shows the patient and the bottom one shows the control. And you can appreciate that the maternal allele in the control is hypomethylated, meaning no methylation. And then the paternal allele is hypermethylated. In the patient sample, you can see both the haplotypes are hypermethylated. And you can immediately put this deletion together with the methylation pattern to recognize that this is actually causing the Kagami-Ogata syndrome. Here's example 2, where we used isoform sequencing, PacBio Iso-Seq, to resolve a variant of uncertain significance. This is a kid came to us with hemolytic anemia in the newborn period, and they required chronic transfusions to maintain hemoglobin level until 6 years of age. They had multiple panels and genome-wide testing. They were all negative, except there was a WUS reported. This is a non-genic upstream variant reported from the chromosomal microarray. And here I'm showing you the short-read genome data. Here's the most commonly used transcript of HK1, and then the duplication is in the upstream region of this HK1, right? HK1 is an interesting gene. Like, it has multiple alternative spliced and tissue-specific transcripts. And you can appreciate, like here I'm trying to orient you with the main transcripts. And here's the transcript that everyone uses, like including the AGMD. And there are multiple other transcripts that you can appreciate. And here's the expression of the GTEx. This particular transcript, specific to testes, but then, there are several other transcripts with differential expression. In this patient, we had to understand, what is going on with this variant, especially in connection to the HK1. So we needed a way to characterize, we needed to be able to reconstruct the transcripts in its full form, and we used PacBio Iso-Seq. And when we did PacBio Iso-Seq in the CD71 enriched RBC cell types from the patient, we were able to immediately see that HKR transcripts, which is very specific to the RBCs, are highly expressed in the patient cell line, versus in the controls, you can see it's non-existent almost. So this gives you the opportunity to reconstruct the full-length transcripts and be able to immediately recognize, and we were able to resolve this duplication as a cost to the patient's phenotype. We talked about many success stories from the research, and now the question is, what is challenging? What are the challenges in bringing long reads to clinic? Cost used to be a factor, but not anymore. But I think annotations and population scale resources are important for bringing long reads to clinic. These efforts are happening worldwide, and I know there are a lot of consortia working on creating these resources, and I hope this will be solved in the next few years. Clinical consensus and standards are evolving again. Clinical labs wait to watch for these standards to evolve, and I think it is slowly happening right now. The evolving landscape, the bioinformatics tools and analytical workflows are also a challenge. But again, this is a field that's constantly innovating and evolving. Throughput is a concern for somebody like us. We need to be able to do X thousand genomes, and we need to be able to have redundant machines in the lab to be able to offer the same kind of testing for all our patients. And we're hoping that this is a minor issue in the next couple of years. In some rare cases, long reads are not long enough, but I think that's that you just have to take it. So now coming back to my original slide about this moving towards a single comprehensive genetic test, I really think long-read sequencing is going to be the single diagnostic test where you can detect sequence and structural variants with the same fidelity, get phasing out of the box for most of the regions in the genome, and methylation right out of the box. This is where the field is moving. I hope to see use long-read sequencing in the clinic. With that, I would like to thank my collaborators and my colleagues in the CHOP Undiagnosed Diseases program and the CHOP Division of Genomic Diagnostics and all the funding sources. Thank you.
Hi, Ramki. All right.
Cool.
Great. Thanks for that talk. We have a few questions. Again, if anyone has questions, please drop them in the questions module. We'll try to get to them. I'll start off with, let's talk about the cases that you went through in the undiagnosed disease program. Many of them have exhausted their standard of care options. How do you decide which undiagnosed samples are best for long-read sequencing? And has that selection criteria shifted as your experience with HiFi has grown?
I think phenotype is the main driver when we think about, there's a biochemical result, there's a metabolic result, and that tells you this is a specific pathway. For example, all those Cornelia de Lange cases, we were confident that they have Cornelia de Lange diagnosis. It's just the variants are missing, right? So I think phenotype has been the primary driver in terms of deciding. Yeah, and if your search space is large, then I think it becomes a little more challenging given the existing workflows. But yes, if you know the phenotype well, I think that's something.
Are there specific patient populations or phenotypes that you think routinely long-read sequencing should be considered earlier? I think we kind of get this question a lot. Where do you first, which ones are more likely to have solves with long reads?
I think anytime you think about these, repeat expansions or segmental, duplication mediated variants, I think those are immediate targets. I would say they're under assayed, to be honest. So I think that's probably the first place I would start thinking. But that's a heterogeneous score. It's not a specific phenotype, but more variant. I would think like that.
There's a related question that just got put in the chat. In your UDN cohorts, what percentage of diagnosis do you think genuinely depended on long-read sequencing because the causal variant would likely not have been adequately detected or resolved by short reads?
So I would say a small percentage, right? Mm-hmm. But then there's a cost to this. It post hoc, you can see, and I can detect this with short reads, and that's been the case for many cases. I would think from the workflow perspective, how long does it take to get to that? For example, there are some cases that are not presented here where we have seen an insertion, but then if you were to depend on the short reads alone, you're looking at 10,000 variants per sample, and predominant of them are noise, right? And to get to that answer, it's very hard. So I think that's what the difference is. If you ask me, the methylation one is a very unique, specific one. Mm-hmm. There are a few cases like that where you need long reads, but if you have sophisticated analysis and them, but then at what cost is the question.
Yep. Maybe I'll do one high-level question, and then we can go into some of these more detailed questions. So I want to talk about multiomics and methylation. I think you give a lot of good examples of research applications in multiomics. When do you think these will be more integrated into routine clinical workflows, or do you think they're still going to be for a while, just focus on research studies in these difficult cases?
I think we're doing multiomics today, I would say, but then there needs to be a strong a DNA variant to get there. I think the methods and tools for interpreting these things together at the same time is evolving. We're not there yet, but a lot of times when we see something, you definitely need, and then there's a follow-up. RNA has been pretty useful in the lab that. And methylation as well, and definitely in the cancer space, there's a lot of methylation application that's already in place. People use it in different for different reasons.
Okay, let's dive into some of these specific questions. We have several questions talking about mitochondria. Could you talk about, let's see, depth of coverage that you are seeing with mitochondria? Do you see mosaicism and heteroplasmy detection? And then somebody's asking about tools to look at mito-DNA. I will call out, we have a PacBio developed tool called MitorSaw, MITORSAW. It's available on our PacBio GitHub. But Ramki, maybe you can speak to your experience looking at mitochondrial DNA, if you have any.
Yeah, so it's similar to short reads, you get mitochondrial DNA sequence out of the box, right? And there are a lot of analytical workflows that's available. MitorSaw is one, and Mito HiFi. There are a few workflows. In fact, we published a workflow from our lab. And in terms of coverage, we're doing, if you do 20 to 30x genome, whole genome, you can expect, I think, anywhere from 7 to 10 times, in the mito genome, and that's good enough to assess heteroplasmy. In fact, we have a publication talking about multiple deletion syndrome in the mitochondria, and it's pretty neat, actually. You can reconstruct these events at breakpoint resolution, which has been very hard because our mito workflow depends on a long-range PCR amplification. Here, we get unamplified. So it's been pretty good. Did I miss anything? It's pretty accurate. We have done some assessments of heteroplasmic level comparison, especially if it's more than 10%, you can be quite confident from PacBio data.
Great. Maybe just one going back to the methylation part. Somebody's asking, are you evaluating methylation signatures and outliers systematically across the entire cohort, or is methylation kind of what you showed more on a case-by-case basis once you have a strong candidate locus?
In our case, I think it's case-by-case basis. we haven't built, so for methylation, tissue type is very important, right? All the methylation signatures that's existing in the public domain is whole blood. But I know there are other labs, we're working on this topic. In our case, it's very specific, case specific. But yes, we have looked at methylation patterns in the LCLs, and they're quite different than what's in blood, and I think there's tissue specificity that you have to take into account.
Yep. I guess this is a question for both RNA and methylation. Somebody's asking, would a targeted approach be more straightforward here? I know we have targeted RNA sequencing. Are you more looking genome-wide, I guess, versus targeted in these situations?
So a lot of times this is still targeted, right? If you want to go unbiased analysis of RNA or methylation, there's a lot of, for example, in the short read, I can speak that the RNA, analysis mostly depends on the outliers, right? People talking about outliers, but then whatever happens elsewhere in the genome can mask the effect that you're looking for. I have a patient we have a patient where we have a gene deletion, ANCOR11, they have a KBG syndrome. But if you look at their outlier expression, it doesn't come out because they have a huge benign duplication elsewhere in the genome, and that masks it, right? So there are a lot of analytical advancement that is required to be able to use RNA as a first line or unbiased analysis. Right now, we're producing examples like where RNA helps, like, we have found answers using RNA outliers, methylation outliers, as from a cohort perspective.
Yep.
But that's not going to fit, like, every one. If you take a single patient, how are you going to do this now? I think that's the big challenge in the clinical lab. And we're not there yet for that kind of analysis.
I have a few questions that are asking about the target coverage that you're aiming for per sample. And then with SPRQ-Nx, the price that I showed, the $350 per genome, is for 20x coverage. How does that coverage trade-off work in the research versus the clinical space? What is sufficient in your experience recalling things like SVs, repeats? And I think it's a trade-off between cost and sensitivity at this point.
Okay. So I think, like, I would like to do 30x, but then there are now data out there. There's a really nice publication that talks about, like, 20x is like good enough to call most of the variant. But then if you want to be comprehensive, probably 30x is a good place to be. And that's math, right? Like, if you want to detect a heterozygous variant, like, we're talking about binomial distribution, and, like, you need 13 reads to call a heterozygous variant confidently, right? So things like that. Yes, like, I think the new chemistry and the increased throughput, reduced costs, I think that should help us do. But, like, 20x is definitely not bad. Like, we've done 20x genomes, we've done like 40x genomes. I think, like, it depends on, like, where you're looking. Yeah.
And then we just have 2 minutes left, I guess. And there was a few questions about, downstream of a diagnosis. Do you guys think about downstream management?
We don't directly. I'm not directly involved in that. Again, I can cite a case. Like, there's a really nice paper from, like, where we collaborated with Danny Miller at University of Washington, and, like, we found this insertion, and the patient became eligible for a particular therapy. But I think getting diagnosis sooner and with less cost, I think that's what I think is. I don't think beyond that, but I think that's going to enable you to take advantage of, like, the treatment or therapy avenues available.
Great. And then I guess just one more question. You've been working with HiFi for a while now. What progress are you most excited about, and where do you look forward to this going in the future?
I think the bioinformatics tools are evolving, but I feel like, that the analysis piece is the most important when we think about bringing long reads to clinic. I think the variant detection is, like, we're getting to the, like, if you think about the sensitivity and specificity, you can get there. I think the tools are already there, but then the tertiary analysis piece is very important. For that, you need, like, large resources. Like, you need to have, like, large databases of variants, false positives. So I think those things are evolving, and I'm excited about all the possibilities with this. I think the tertiary analysis piece, if that comes together, I think, it's great time.
Fantastic. All right, so we're at time. So with that, we'll end there, and any questions we didn't get to, we will try to answer offline. Thanks, Ramki, and thanks everyone for attending.
Thank you.